Good morning, good afternoon, good evening. Let us go ahead and get started with day number 4 of the May Institute on Computation and Statistics for Mass Spectrometry and Proteomics. It is very nice to see everybody joining us again. I hope that this will be a useful experience for you in these 2 weeks. This is the first time we're doing this online format, and a lot of this is quite new for everyone. And so one thing which is definitely new this time is trying to do some kind of a hands-on experience remotely like this, where we can try to share the specific skills or specific functionalities of a tool with all of you so that you can do the same thing in your own work and you can apply the same skills with you. So this is a experimental session for us, and I hope that it will go very well. So before we start, let me just show you some logistics. At the bottom of your screen, you have 2 buttons. There is a chat box and a Q&A box. So the chat box is for you to communicate with the panelists if you have some logistics questions, for example, if you need to find a URL or if you need to find some other information. But if you have technical questions regarding the presentation, please use the Q&A box, because the Q&A allows us to better manage all the questions and answer them in a different time. So this is what we're going to do. So let me now introduce our speaker for today. It is my great pleasure to introduce Brandon MacLean from University of Washington in Seattle. So Brandon is a computer scientist. He worked as a software engineer and developer at Microsoft for 8 years. And during this time, he was involved in the Visual C++ Developer Studio project and multiple other projects. And I think from this experience, Brandon brought an extreme rigor and tremendous skills of software development and software validation and developing projects that are functional, robust, and also easy to use. And so after leaving Microsoft, Brandon was involved in multiple startup companies. So he was vice president of engineering for Westside Corporation, and he co-founded LabKey Software. So this is a company that you may be familiar with. It provides workflow integrations for lab scientists. And after that, in 2008, Brandon joined University of Washington, the MacOS lab, and started working on software for targeted mass spectrometry, targeted proteomics at that time in the context of CPTAC studies. And this work changed the world of targeted proteomics as we know it, because Skyline came out of this project and it continues to grow in ways which are truly impressive. And so I'm really happy that Brandon agreed to join us today and tomorrow and share his work with Skyline and hopefully teach you the skills that you can use in your own work. Thank you so much and welcome, Brandon.
Hi, thanks for the generous introduction, Olga, and the opportunity to carry on with the May Institute Northeastern. Usually I get to travel to Boston and see some local friends. I was actually born in Boston. But so here I am in Seattle at home, and I assume most of you are at home as well. Welcome. I'm going to try to, Skyline has its own sort of webinar series. We're up to webinar 19 now, and of course, as research scientists, we all know how to present a set of PowerPoint slides. I'm going to try to get a little closer to the experience of being in a classroom with people. Usually, we have a week-long or at the May Institute, it's broken into 2.5 days. 2.5 day sessions, but we get a group of around 50 people and work closely with them and have them do some exercises. So the first thing, well, actually, let me just move to my agenda from my picture here. So the agenda for today, this is my only slide. So we're gonna deal with Skyline itself, and I'm gonna play you one of those webinars I just mentioned. So we're going to start with some Q. We're going to start right off with Q&A because there was something that if you went to the page, you found out there was maybe homework. But I'm going to start with the Q&A is going to start with me asking you some questions, which is pretty typical for our presentations at these courses. So I'm going to start with I'm launching a poll, and I'm going to start. I would like everybody to vote and let me get a sense of your experience level with Skyline before you registered for this class. So we'll see. So we have already 174 participants. Hopefully everybody will answer. Everybody will be able to answer this. It's pretty simple. All right. Well, that is pretty interesting. I'm going to give you a little bit more chance. We'll see. This will give us a sense of how many people actually are paying attention to their screens versus just have this minimized. Okay, there are 30 more people out there. And I guess we'll go with that. Okay, so I'm going to end polling. Seems like most people who are going to say anything have said something. And here we have it. The winner has people had before registering for this, people had used Skyline. A lot of people had used it for SRM and PRM. Fewer had used it for DIA, and then there's a number of people who'd never used it before and never heard of it before. And so used to be that when we did these, about 2/3 of the participants had never even heard of it before. So obviously in the past 7 years, a lot more people have heard of Skyline. So I'm going to move to, I will stop sharing those results, and we're going to move to our next poll, which is your overall DIA experience. So please vote now on this. All right. This is so interesting. Thank you for doing this. Wow, that vote was much quicker this time. We're already up above what we had last time. So I'm just gonna, wow, yeah, okay. Great job, 85%, that's enough to give us a statistical sense. We'll end polling and share the results. And yeah, that's kind of what I expected. A lot of, you can feel really great listening to lectures from people like Brian Searle and Rudy Aebersold. And wow, I understand this concept. And then far fewer people have actually done an experiment. I guess actually in this case, we have 25% of the people have done it and have done at least one. And then we have 10% using it regularly, and a very few people who have never heard of it, but just as many people are totally confused by it as as have actually done an experiment. So that's we'll switch to the next question, which is, how much session prep did you do? So, you know, they're on the you were supposed to do a tutorial, and it looks like a lot of people have just shown up, which is, I kind of expected that. And I'll show you the results in just a moment. So this is really showing the benefit of doing a session live versus doing a webinar, because yep, we're up to our same level, and here are the results. So no preparation at all, got Skyline installed, and then a very small, yeah, actually completed, 5% completed the tutorial, started the tutorial but got stuck, shared, started the tutorial but ran out of time. Okay, so and then just so, yeah. So you're not actually super well prepared. And that I kind of suspected that, which shows us the benefit of having people show up and be in a room, because we would then give you time to do the tutorial. We're not going to have the chance to do that here. But yeah, I'm going to quickly switch to the tutorial that I had asked everybody to do. So this is actually, we're going to have a look at this. I'm going to stop sharing results for the moment. So this is a webinar that we're going to have a look at. There's an associated tutorial, step-by-step written tutorial with data that you can download. And this was the hope was that you would sit down and do this. And at the May Institute, we give you time. The nice thing is that we're sitting around and we can answer questions as you run into them and keep you going. So the people who said, oh, I started, but I got stuck. And we would try to give just about everybody time to get through the tutorial. So this is a tutorial you can choose from QExactive or TripleTOF data. We're going to be focused on the TripleTOF data ourselves today. But I highly encourage you to actually go back and do this tutorial. It's a really important part of understanding things. And I would say that hopefully people will be interested in attending the session tomorrow. But before you do, it would be great if you could sit down. This may take you an hour or two to get through, but it's well worth struggling through and actually doing the hands-on yourself. So not many people had done that yet. So we'll get back to this. I think actually my next question will all right, so now this is a webinar that we did a few weeks ago. It really covers a lot of the content. So this tutorial is all, we had a tutorial that we're using for a couple of years created by Ben Collins, and we just completely repurposed it and updated it to the latest Skyline. And this is the tutorial we would have used in Boston had we had the May Institute there. And then we also, Ben and I did a webinar, a Skyline tutorial webinar. This is number 18. You can get to a very extensive collection of these webinars. If you go to the place where you installed Skyline, hopefully most people installed Skyline or have Skyline installed. There's a webinars link over here under pages, and you get the full collection of all 18 webinars that we've done since late 2014. And we've just done 2. And today we're going to focus on this one, which is capturing a lot of information that we would have presented at the May Institute. I mean, when we started this curriculum, it was Ben and I, and then Brian Searle stepped in to take over for Ben when he couldn't make it. But so my next question is, I'm going to play this webinar as the next step, and then eventually we'll get to we're going to go beyond what this webinar shows. But I want to get a sense from you, the audience, where you want to start. So so I'm polling again. I like your input. Do you want to watch the full overview from Ben Collins, which will give us some perspective on the data, and maybe be some review of some of the concepts that Brian Searle presented on Tuesday, and then some people may be like, well, I saw this already. I really just came because I saw that you mentioned PROSIT. So I like to get a sense of where everybody falls on that one. And we will, for those of you who only came for PROSIT, you probably can come back in an hour, I think, because you're not the majority. I can already see that. So, all right. So anybody else want to vote here? Getting close to 140, which seemed to be about our, well, a few more people have joined. Okay, that's good enough. End polling. So we're going to start from the beginning. For those of you who really feel like you understand DIA and hope to jump ahead 15 minutes, hopefully you can bear with us. And for those of you who really have seen this webinar already and really want the follow-on material, I would say, you know, you can leave your sound on for an hour and do something else if you want. So let's just, we're going to go to this webinar here. Good morning, this is Brendan MacLean, and I'm gonna switch to this is, I mean, you can get to this recording too if you want to re-see this, and we're gonna switch to this location where it's very close to the start of Ben's introductory lecture. And
okay, super. Thanks a lot to Brendan and Nat for having me on today. So here's a quick outline of what I'd like to talk about today, and there's basically three topics that I want to touch on. I want to just briefly mention what's the idea behind DIA or swath type approaches with respect to acquisition. Second, I want to talk about the kind of concept behind the data analysis that we're going to be using today, so-called peptide-centric or targeted data analysis. And then third, I just want to mention the data set for the tutorial and where this came from. So this was originally inspired by the LFQ bench data set paper from Pedro Navarro and colleagues. So let's start with DIA. So this is a figure from a tutorial focused on DIA and swath type approaches, which tried to capture some of the diversity in DIA methods at the time we wrote that in 2018. And so it's sorted here by year of publication from bottom to top and by precursor isolation width from left to right. And so I think the first thing to notice here is that DIA and proteomics has been in development for a long time, starting with these papers from John Yates and people like Dave Goodlett back in the early 2000s, running up to things like swath and beyond, which we worked on for many years now. And I think the point of showing this figure is really to emphasize the diversity of methods out there that fall under the umbrella of data independent acquisition. And I think there continues to be a lot of development in this area. But another thing to say is that several of these methods have now really matured into techniques that are being routinely used in applied projects. And there seems to be a lot of momentum in this space in the last 2 to 3 years. That's my sense of groups that might have previously used methods like DDA, making the choice to switch to some kind of DIA-based method, which I find actually very encouraging. And I think the biggest element that's helping to facilitate this transition are really the development of strong computational methods for DIA data analysis from many different groups, and also in particular the accessibility of those methods. And I think that Skyline has had a big impact there, and I think will continue to do so. So, okay, so what does a DIA method actually typically look like? So here's shown schematically what a typical method could look like in terms of the duty cycle on the top there. So usually we have a precursor scan, which is followed by a number of MS2 scans using a wide precursor isolation window. And in this case, this method is actually basically the sort of classical swath 32 windows times 25 MZ method. And the thing to notice at the top here is that we essentially cover the precursor space in a comprehensive way with MS2 spectra in this space where we expect tryptic peptides to be. And so we should basically be capturing everything that there is there to capture. That's basically the main idea behind DIA, I would say. So if you look in the MS1, the precursor spectrum on the lower left, you can see that one of these 32 precursor isolation windows here is highlighted. So the window from 925 to 950. And in that window, it looks like there are 3 major precursors highlighted in blue, red, and green. Of course, there could be much more than 3, but just 3 in this case. And so when we move to the MS2 scan on the lower right, basically what we're doing is co-isolating and co-fragmenting these precursors. And the resulting spectrum that we get is shown in the lower right panel. And I think the thing to notice about this spectrum is that it's very complex, right? So we have fragment ions from all 3 precursors, and it's going to be very difficult to interpret this spectrum in a kind of classical way. And so I think this is really the key thing to understand about most kinds of DIA data. So that is that you can get comprehensive coverage of the precursor space with MS2 spectra, but it comes at a cost, and that cost is that the spectral complexity goes way up and the data is more difficult to interpret. And so in the interest of time, I'm going to move on here, but if you're kind of new to the DIA world, then I think this tutorial paper that we wrote from in 2018, led by Tina Ludwig in molecular systems biology, could be a good starting point to learn more. So let's talk about the data analysis concept. So just to illustrate this kind of spectral complexity point a bit more, this is what a real spectrum looks like, a swath MS2 spectrum from the precursor isolation window from 600 to 625. And you can see that the spectrum is pretty busy, right? And so the peaks highlighted in these, the peaks highlighted in color there are from this particular peptide sequence in pink, which I happen to know in there. And what you can see is that so these are the major fragmentines which are of this peptide which are being highlighted, but they're not the major fragmentines in the spectrum, right? So they're quite down in the grass. You can barely see them. So this spectrum is going to be very hard to interpret in a way that retrieves this peptide, right? And so you can imagine if you knew something in advance about this peptide and how it behaves in the mass spectrometer, then maybe you would be able to dig that signal out just by focusing on the places where we would expect the signal to be. And so the solution that we and others have used for this problem for a while now is to ask this question in a sort of targeted way, or a sort of peptide-centric way. So the question is this peptide present in my data, or do I have evidence to support that? And we can make this an easier question to ask by building in some prior knowledge about what the peptide looks like, right, to the mass spectrometer in the form of spectral libraries. So here's again one of these complex looking swath or DIA style MS2 spectra, and if we now include the library information for this particular peptide. So in the mirror plot there, you see the major fragmentines for this particular peptide that we're interested in. Then we can sort of test this hypothesis. So let's say we have a hypothesis that this particular peptide might be present in the sample, and we can ask the question, can we find evidence for the presence of this peptide? So let's say we zoom in and say, are there particular fragmentines that we expect, right? So this is now the Y5 fragmentine that I've zoomed in on, where we see an isotopic envelope where we would expect it to be. So that's good. So let's draw a box around that, and we're going to extract an ion chromatogram. So we're going to plot the intensity of this Y5 fragmentine as a function of chromatographic time, and we're also going to plot all its friends, all the other fragmentines that we're expecting to see from the library. And now we get these traces here shown in the middle panel. It kind of starts to look something like SRM data, and we see a signal group, a peak group in the middle, which looks like it might be our peptide. And so at this point, what we need is some kind of objective way to compute scores and to say whether this really is the peptide that we think it might be. So and to calculate the false discovery rate. And so what's shown in this lower left panel is the kind of the M-profit type of algorithm, where we're going to compute all these scores. Like, we're going to ask the question, do the fragmentines all coalute with each other nicely? Are there fragmentine intensities, the relative fragmentine intensities consistent with what we have from the library and many other scores? And we have a method to integrate these and then try to compute the false discovery rate. And so this picture is, in a nutshell, is this sort of peptide-centric or targeted data analysis concept. And this is essentially what Brennan is going to be doing today in the form of Skyline in the tutorial. Okay, so let's talk about the actual data set for the tutorial and where it came from. So as I mentioned, let me say first, so for quite a few years, we've been running courses on targeted proteomics at ETH Zurich, with more recent courses actually focused on DIA in 2017 and 18. And we haven't run since 2018 because Rudy Aebersold was starting to close his lab in 2019. But actually, I wanted to mention that I'm thinking of setting this up again in my institution in Belfast, possibly next year if things calm down a little bit. So if people would be interested in that, they can feel free to shoot me an email and express interest there, and that might get me motivated to try and get this thing going again. And I want to say that there's actually been a great relationship between these courses that we ran in Zurich, along with a whole raft of other courses taking place worldwide. applied on related topics. And I think Brendan has really been the backbone of this kind of broader effort to run these courses. So incidentally, I just want to mention that if you're interested, we have a YouTube channel with the videos from the last roughly 4 years, I think, of these Zurich courses, if you would looking for more resources. And the reason I wanted to mention these courses is because this is the original source of the data set that we're going to use in the tutorial today. In particular, this 2017 and 18 iterations of the course, we acquired these new data sets for the purpose of tutorial purposes. And Brendan and his team have recently been updating this tutorial to include all of the latest Skyline kind of functionality. And this is what he's going to be demonstrating today. Okay. So now I want to introduce the study that kind of inspired this data set that we're going to use for the tutorial. So this was a study coordinated by Pedro Navarro and Stefan Tenzer, and they wanted to ask the question whether the tools that were the data analysis tools that were around at the time for doing DIA analysis were producing kind of comparable results. And in order to do that in a kind of a fair way, they needed to have some kind of ground truth sample that they could compare to. And so their solution was to use to analyze protein extracts from 3 different species. So this is E. coli, yeast, and human, but these were mixed together in defined ratios in 2 different samples, so that they'd have a ground truth benchmark to kind of benchmark the data analysis against. And so this is depicted in these kind of tubes marked A and B on the on the left panel, showing what the fraction of each of these species was. And so what they did was they acquired these samples in swath mode on triple TOF instrumentation, and then they gave the data out to the software developers for these 5 tools: OpenSwath, Swath 2.0 from Sykes, Skyline, SpectrumRod, and DI Empire. And they said, well, analyze the data. And then they had a round of kind of iteration where the developers were able to talk to each other and kind of make improvements in their tools based on comparative analysis. And this actually turned out to be, I think, a very useful exercise. And I think all of the tools were improved based on this kind of benchmarking study. So let's take a look at what some of the typical data should look like. And this is, again, data from this Navarro paper. A typical output looks like, now just showing the results for 2 tools. So this is OpenSwath and Skyline. And for these plots, the way to read these is on the x-axis, you have the abundance for inferred proteins from these samples. And on the y-axis, we have the log2 ratio between the A and the B samples, right? And so the green cloud are the human proteins, and you can see that these are basically centered on a log ratio of 0, so no change, which is as it should be. And the orange and the purple are yeast and E. coli, which are basically you can see these clouds which are centered at the expected ratio, save for some compression in the lower abundance range, I would say. And the difference between the left and the right panels is this sort of before and after this step where the developers had the chance to kind of improve the tools. And you can see that actually this made a significant difference in the quantification, especially the ratio compression in the lower end. I also think it's nice to see that looking top to bottom, so comparing the 2 software tools, we get actually quite similar results. And so the questions are, well, the first one I already mentioned, we want to ask the question, are the quantitative ratios, as expected, which they broadly speaking are. And second is, the second is less obvious, sometimes overlooked, but are there a lot of data points out of position with respect to where they should be in terms of the species? Like, i.e., are there purple dots up in the orange cloud? Are there orange dots in the purple cloud and mixed in with the green, and so on? And this would really probably indicate a problem with the false discovery rate in terms of assigning the peptides and proteins. And so there are a few of these, but they're actually relatively few compared to the number of dots that are actually on the plot. And this gives us some confidence that we're not doing something too crazy with respect to the false discovery rate assignment. Okay, and I also want to point out that in the last few years, this approach has become a popular way to do benchmarking of new methods. And papers like when people are writing manuscripts, they very often like to include something like this. And if they don't, the reviewers sometimes like to make them include it to convince them that their method is working in the respects that I mentioned in the last slide. So here I show a figure from, actually this is a preprint that we put up on DI Passive, recently where we used a similar approach, this time with just 2 species, yeast and human, but it's essentially the same idea. And I think if you look at other recent papers focused on method development for quantitative proteomics, and especially in the DIA space, you'll see that, and this can be either from the software perspective or from the data acquisition perspective, you'll see a lot of these kinds of figures. And this LFQ bench style experiment is becoming, I would say, almost becoming standard practice for benchmarking new methods. Okay, so let's look at the data set for the webinar. For today's tutorial, I should say, and this is essentially very similar to the data from the LFQ bench. So it's a reconstituted version of the mixture from the LFQ bench Navarro paper. So we have expected ratios of 1:4 in E. coli, 2:1 in yeast, and 1:1 in human. And incidentally, just to note that I got actually the AB labeling is opposite from the Navarro paper, and this is just my fault. I wasn't paying attention, but in case you're wondering why that is, we injected this in triplicate in DIA mode, either on a 6600 triple TOF or a QE plus using a 1-hour gradient. I think Brendan is going to focus on the triple TOF data for today, but we also injected each sample once in DDA mode, each of the A and B samples, for the purpose of building a library. And this is not going to be a especially deep library, but it's perfectly fine for teaching purposes, I think. Okay, and so with that, I think I'll pass it back over to Brendan. Thanks for your attention, and I'll be back after the demo for the Q&A. So, Brendan.
All right. Well, thanks, Ben. That was a great introduction, and I'm going to switch back to my screen here. So I'm going to try to get started right into the Skyline walkthrough as quickly as possible, and reiterate that the intention of these, the reason we call them Skyline tutorial webinars is we're hoping not just to show you a bunch of stuff, but to get you started and using and hands-on learning the techniques and using Skyline. As Ben said, this was originally a written tutorial which was presented at the DI SWATH course, and we've now just gone through a fairly extensive revision for both of these data sets. You can see this page on the Skyline website is up there now, and it'll be linked to from this webinar page, and you can see that there's sort of extensive links back to this current webinar and webinars we've given in the past. So this is all ready to go, and when you're finished, you can go back and do the written webinar, or you can watch the recording of this and follow along with me what I'm doing, which will not be as complete as the written webinar. But so now we're switching. I'll get rid of this view, well, Skyline will be up in just a moment. So in the original tutorial, when I went to ETH, it started with clicking on the blank document, and then there were quite a few complicated steps to prepare a document and actually execute this particular DIA analysis. And I went back home with a lot of notes on how to improve that, and Kaipo Tamura, who I think is probably listening at this point, did a really wonderful job of improving this import DIA peptide search wizard. So we're going to use that today. Actually, before we get into that, let's quickly take a look at the data set itself. This is what the tutorial, if you do the written tutorial, what it tells you to do is put one of these data sets, extract it from a zip file into your documents directory. Here is the triple TOF data, as Ben mentioned, in my documents directory. If you're more interested in the QExactive, there's a companion data set for that, or you can do both. So once you drill in, you can see there's a DDA search folder and a DIA folder. We're going to start in the DDA search folder, which has everything has been converted to MZ XML or MZML from the raw data formats, just because that was for size. They're all centroided. And then these PEP XMLs are the individual search results, which I think are done with Comet. You can see the Comet params here. And then run through the TPP to get peptide profit probabilities assigned. And then this is the roll-up, this interact PEP XML. And then down here at the bottom is the full FASTA file. This is exactly the same FASTA file that was used in that paper that Ben mentioned from Navarro. You can see N.A. Pedro, Pedro Navarro, that's his name. So anyway, and then it's 3 organisms with IRTs spiked in, and then it's got forward and reverse. So that's what that FASTA file is. That's what we're going to use today. And then in the DIA folder, we've got the 6 runs in MZML format, again, centroided, and they were originally WIF files in this case from the triple TOF, and we have the A's and the B's. We also have this target FASTA, which is the tutorial will walk you through using the target FASTA. It's usually what we teach at these week-long courses because it's smaller and faster and everybody shows up with laptops. Today I'm going to be working on a desktop i7 computer with a hex core. So that's going to allow me to import all 6 MZML files in parallel. So it's going to be faster, and hopefully that'll keep you from waiting too long. That's the data. We're going to get started by clicking import DIA peptide search, and it says, well, I need to give it a name. I'm going to click OK, and I'm going to go back up to this root DIA TTOF folder, and I'll call this DIA TTOF. So that's my file name. I put it in the root. Now Skyline asks me, okay, where are the files that you're going to build into your spectral library? And it also asks me for a cutoff score. And this cutoff score is sort of normalized to the peptide profit scoring probability orientation, which means that 0.95, it's closest to 1 is best. So those are the peptide profit scores. But if you're using a search engine like MaxQuant or something, where or you're using Percolator with Mascot, then the score that you may be used to thinking of is a Q value or P value where 0 is the best, and Skyline just flips that by subtracting 1. So if you're using something with a Q value or FDR, you might choose 0.99 to get a 1% FDR. Since peptide profit uses posterior error probabilities, we can get better than a 1% FDR with 0.95. So that's what I'm going to use here. Trust me, it ends up being better than a 1% false discovery rate, and that's maybe something that you work out yourself with your own data and search engine. So now we're going to go and add a file by going to this DIA or this DDA search. And as I said, these are the individual searches, and this is the roll up with the probability that I'm interested in. So I'm going to choose interact PEP XML. And then down here, this is one of the things that Kaipo added, and it's actually done quite a bit of work on, is this IRT standard peptides, just being able to choose them and having the IRTs get built into the library. And in this case, Ben has spiked in the diagnosis kit, and we're going to use all 11 peptides. We also get to make the choice of whether or not to include ambiguous matches, and that means whether or not we're going to include things that the search engine identified, where it identified multiple peptides in a single spectrum. And we're really interested in getting canonical spectra, in this case, to use in our DIA data. If we were doing MS1 filtering from DDA, we might choose this, but in this case, we'll leave that unchecked. So next. So that starts Skyline building the peptide search library. Which should go pretty quickly. This is an area where we continue to put in a fair amount of performance work, thanks to both Kaipo and Matt Chambers of the Skyline team. And so this shouldn't take very long. It's reading spectra out of the MZXML files. It reads the search results out of the PEP XML, and then finally it creates a non-redundant library. And Then it does regression based on the IRT peptides. It looks like it's found all 11 IRT peptides. It was able to do a regression with a 1.0 correlation. And then this is the slope and intercept. And if you click on this success link, you can see that regression. See, it's quite good. Can we? No, we can't. But you can see also that these points are not exactly on this line. These are the original IRT calibration values supplied by Biognosys itself, and most of these kits will have a supplied IRT value within this IRT C18 range. So that's the IRT values are over on the left, and then these are the actual measured retention times in the DDA. So pretty much on the line with a really high correlation, but not totally on the line. And so when we click OK, Skyline will say, hey, do you want to recalibrate those IRT values to be exactly on that line? And if you're planning to reuse this IRT library that comes out of this, you might say no. If it's really just a single data set library like ours is today, then you can say yes, because that will tend to give you a better regression because it's based on exactly your own chromatography, rather than the original IRT values are based on how the chromatography and the calibration done by Biognosys. So we prefer to recalibrate to our own chromatography here. Here, Skyline just lists a bunch of peptides that were identified in ambiguously in spectra. So that means that from a spectrum, single spectrum, the search engine comment has said, well, these are the two peptides I think that are in this spectrum, and we prefer to have canonical individual spectra, which we can hope for in DDA, not so much in DIA as Ben explained. So just click OK. At this point, if you have ever done the MS1 filtering in Skyline, usually Skyline will just find the MS1 files to extract chromatograms for, but in this case, we're now extracting chromatograms not from the DDA data, but from the DIA data. So we have to tell Skyline where those are, and they are here in this DIA folder. And then my favorite way to choose all of them is just to click and drag a box until they're all blue and click open. So there they all, am going to leave this default. This is now the default, which is many simultaneously, which is going to allow Skyline to import all 6 of these in parallel. And now it tells me that, hey, there's this long prefix on the beginning. Do you want to just remove that and have these shorter names? And that ends up leading to better display in Skyline. Certainly for the summary graphs, we have to show these names in the y-axis category, or in the x-axis. The categories for the x-axis will show up as these names, and if we have this big long name, it won't show up as nicely. So I'm going to accept that to remove it. Next, I'm asked, what I want for precursor charges. I'm going to go ahead and add 4. Certainly, if I have precursor charges 3 and 4, I should have fragment ion charges 1 and 2. I'm going to remove the precursor here. Skyline can allow me to extract not only Y and B ions from the DIA MS-MS spectra, but also precursor ions from the MS1 spectra, and that can add interesting information. There are certain modification types that are hard to tell apart if you don't include the precursors. So there's reasons for that, but it would make the experiment a little bit bigger today and a little bit more complicated to analyze. So I'm just going to leave that off. Here I'm going to check use DI precursor window for exclusion. This is something that was brought to my attention by the Aebersold lab and actually implemented by Hannes Roos, the owner of OpenSwath, to say that we don't want to include Y and B ions that actually fall within that range that we're filtering, because a lot of precursor ions are flowing through to our spectrum from the precursor range that we're isolating. The ion match tolerance of 0.05 default here is pretty good. That ends up being about 50 ppm, which is great for a triple TOF. And then this 6 product ions, that's what was suggested in the original open swath manuscript. And certainly I know what Ben and colleagues at ETH use a lot. It seems to work well. And then you get to decide whether or not you want to take only things that have 6 ions. There may be some spectra where you can't necessarily get 6 ions with these filtering criteria, and you could loosen this up to 4. But we're going to keep it fairly stringent and say, no, if you didn't have 6, then we're not confident enough in that measurement that we're going to use it. So this will filter out some of our match spectra. Next, now here you can see Skyline is saying, hey, I'm not going to do any MS1 filtering because you didn't include a precursor ion, which is what we want. And then it says by default, okay, DIA from centroided mass analyzer, and that's what we'll use because we have those centroided MZML files. If we actually had native WIF files, I would say that we would want to choose the TOF option. And when we do that, you can see it switches to a resolving power rather than a ppm. So, but we're going back to the centroided. And then I happen to know ahead of time, and Ben included in his original instructions to set the mass accuracy to 20 ppm. We're going to take a look at the mass accuracy and the mass error in this data, and maybe we'll get some insight into why 20 ppm is better than 10 ppm. And as an exercise for you, if you end up working with this data, you might actually try both and see how they work. I also know that this data has really pretty incredible chromatography, and we could reduce the extraction window. This is plus or minus 5 minutes would be a 10-minute extraction window around the predicted retention time. We could reduce that to 3, maybe even 2, but I'm going to leave the default, and we'll look at that once we get the data imported as well. Oh, that's right. So I need to actually define an isolation scheme. I can say results only isolation scheme, but I guess it's probably complaining about the fact that I used I said I wanted to use the filter windows as an exclusion. If I use results only, then that prevents me from having Skyline know a priori some things about what targets I can measure and which fragments I want to measure. And so Skyline is capable of looking at the data and figuring out what windows to extract from. But it's usually better to predefine your isolation scheme. So that's what we're going to do here. Click add, and then name this TTOF 64 var. So that's it's 64 variable window TTOF. And then this is use results. These data, as I think Ben described, have a margin, so you could actually use results with a margin, but we're going to do pre-specified windows. And after a lot of sort of back and forth communicating with people who are giving me data and trying to figure out what their isolation schemes were, and sometimes people getting their own isolation schemes wrong, I finally implemented this import button, which allows me to just go to a file, and we're going to pick this first one up at the top here. It could be any one of them. They all have the same isolation scheme, thankfully. And then Skyline can read that isolation scheme pretty quickly and even figure out that there's a 0.5 margin, because these windows, so you can see the measurement windows 408 to 407.2, they have a 1 MZ overlap. And you could actually, well, let's look at it in a graph. So if we click on the graph, and I can actually maximize that, you can see this is the same isolation scheme repeated 3 times. So cycle 0, 1, 2, and you can see there's a little bit of pink and there's blue. The blue is the extraction range, and the pinkish color is those are the margins, and I can click this checkbox, remove the margins, and now you can see there's no overlap in where we're going to extract. So we've measured with some overlap, but anytime we get a precursor, we can say definitively what spectrum it was measured by. So that's how that works. We're happy to see that there's no red or yellow. If there are red or yellow in this graph, that would indicate a problem. So that graph can be very helpful. That's all we need to do for the isolation scheme. And now we can see that that's all set up, and then click next. So finally, we need to tell Skyline what we're going to target and whether or not we're going to allow miscleavages. Again, you could. We certainly do in some experiments, and those will be backed by what's in the library. So it's only going to include things that are in the library. But today, I'm going through at a fairly minimal level to get a really high quality data set. So I'm just going to leave miscleavages at 0. And then, as I say, we're going to browse, and I explain that we're going to use this whole proteome. We're doing proteome-wide DIA today. The written tutorial itself will take you through targeted DIA, but this is going to be proteome-wide. So click open, and then here you can see, so that's that FASTA file. And then now we get to choose. This is more great work by Kaipo Tamura. You can actually choose decoys. So in order to actually run that whole statistical analysis that mProfit that Ben mentioned, we need to have decoys, and we get to tell Skyline how we want to produce those decoys. We'll use shuffle sequence. We can even tell Skyline to use how many decoys per target to use. And by far the default is just to use 1-to-1, but when you get into these really large proteome-wide libraries like Ben's Pan-Human library, with Ben's Pan-Human library, I tended to use a ratio of 0.25. So that means one decoy for every 4 targets. In today's, we don't need that for today's tutorial, so we'll just leave it at 1. And then finally, with this having been set, we can say we want to automatically train an mProfit model and have it applied. So that's that. Click finish. Now Skyline is going through that FASTA file. You can see that sometimes there are proteins called reverse underscore, so it's a mix of forward and reverse. Hopefully we don't have a whole lot of spectra matched to the reverse peptides, but that would be our false discovery rate in the DDA search. After the analysis, Skyline says, okay, it can find spectra for peptides matching 4,908 proteins. If you think about the full proteome of those 3 organisms, it's certainly quite a lot more proteins than that, but this is what the peptide search found. And then this many peptides, so some of these proteins have very many peptides because it's quite a multiple of about 7 or 8. And then some of the peptides have multiple precursors, but not that many. And then finally, because we're using 6 transitions per precursor, we have almost 250,000 transitions. Here, because I know that false discovery rates in DDA are higher among the single peptide proteins, and just to reduce the size of what we do today, I'm gonna limit myself to 2 peptides per protein. So I'm not going to take proteins that don't have at least 2 peptides. And you can see that loses me 1,900 proteins. So fully, well over a third of my proteins have only 1 peptide. And I guarantee you that we have a higher false discovery rate in that set, and that if you use that set for quantification, you're going to have a higher quantitative false discovery rate So here, just to get the cleanest possible data, we're going to just exclude those. You're welcome to use them in your own experiments. And then we're also going to simplify the analysis by removing duplicate peptides, which means, and we lose another 5,000 or so proteins because of that. But it makes the analysis much simpler. We don't have to have protein groups. We can depend on the fact that every time we see a peptide, it belongs to a certain protein. So that reduces our transitions, and it's going to reduce the analysis time today. But you don't have to make these choices. They're just they're gonna end up giving us better quality data and be processed more quickly, which is of interest since you're all watching me. So Skyline is off and running, and you can see, as I predicted, it's actually analyzing all 6 files in parallel. And this first pass is it extracting just the IRT peptides, so those landmark peptides, so that when it comes back and extracts everything else, which is almost 200,000 transitions, it can extract them only in the retention times where they're expected. So it doesn't have to do full gradient chromatogram extraction for everything. I'm going to push this push pin down so that when we're done, it will tell us how long it took to get through all these data. I'm going to go ahead and hide this and show you that Skyline is pretty multi-threaded. So click hide. You can see the status continues down here. Things have actually gotten fast enough that I have to be a little quick to show you all this. And so it's making its progress here. If I ever want to see that graph again, I just have to click this button. And I can certainly leave this graph up, maybe down here, while I begin to review some of these targets. So here's this long targets list that Skyline has created with almost 2,500 proteins, and there are 2 special proteins. One at the top is the IRT protein, and you can see the 11 IRT peptides, and they all have a little clock face indicating that they're related to time and timing. And then down at the bottom, we have just all of the decoys, which are half of the proteins here, just all shoved into one, or half the peptides all pushed into one protein, hidden. So I can click on these IRT peptides to get a sense, and now actually all the import has completed in just 2 minutes. And so, yeah, that's pretty quick. And yes, much faster than we usually get through on a laptop at these in-person courses. And you can see, now it's going to give us so we're on to the M-Profit model. Here is the M-Profit model. You can see that it's all fragment ion scoring. We would have a few more scores if we'd include the precursors, but these are all the scores that are applicable to fragment ion only DIA analysis. And you can see, when you see this, you should feel fairly good because, all of them are enabled, they're all in use. And then you can review the weights of them. So intensity is a good score here, probably because we're using the fragment ion spectra only, and that would indicate that the amount of signal that we're seeing on those fragment ion spectra is higher among our targets than our decoys. Retention time difference is a nice strong score, and that's probably because I included that full 10-minute window. And we'll look at that more closely, but that should make us feel good that retention time is giving us a fair amount of weight. And then, any model that doesn't have the library intensity dot product is a pretty substantial weight. I would really question. The shape score is not providing much value here, and is in fact reversed of what we expect. That's why it's red. Probably not a problem. Maybe it's very similar to some other score, and they're just kind of, you know, it's getting that negative. You could, because it's going opposite of expected, you could drop it and retrain the model. Because generally we want to include only scores that have a sort of have some basis in meaning. Like you don't like, so, you know, if retention time were negative, then that would mean the farther away you get from my prediction, the better. And that's not really a great thing to weight into your model. Coelution, something, and then this coelution count score, which is sort of this score I came up with originally for Skyline, does seem to usually have a decent weight. This was the original Skyline score before we did full M-profit models. And then these other scores, product mass error, you get a little bit of value out of. So that's a great model. No problems. If we look over to the right, then you can see this is the normal decoy distribution is the density plot in the back, and then the histogram plots show us the actual decoy histogram with the target histogram. We can see there's pretty nice separation, and that we've actually really found most of the things we were looking for. There's not a big, you know, if you use one of these proteome-wide pan-human type libraries, then you expect to see a bunch of targets that just weren't even there. But since we're using a dataset specific library, we tend to expect to be able to find most things. So if that looks all good, click OK. Again, it took us 2 minutes to get through that. We can close that status. You could look at each one of them. It's kind of a jumble at this point. So click close. So back to, we were looking at the IRTs, and you can see that the IRTs get extracted pretty much full gradient. And you know, for the what data is there, it seems like maybe Ben cut off some of the spectra at the beginning, but they're full gradient. If we want to look at more closely at the peaks, we can go view auto zoom best peak, which notice says F11 is the keyboard shortcut, and then to go back to none is Shift F11. I tell people that they should memorize those at the courses so that I can go now back and forth between these two views, zoomed in and not zoomed in. I'm also going to, so if I expand this, you can see each one of these peptides has a library spectrum and a really great score. 1.0 is the best possible score. So really high confidence that we found those IRT peptides, which is what we're hoping for. If you click on the individual transitions, they get highlighted here. You can see there's 7 points across the peak gets labeled when I select transitions in the chromatogram plot. You can see them highlighted in the library plot as well. And so, yeah, but so you can kind of review the library plot and see how well you thought Skyline chose things. But for the most part, we're done with the library plot here. And so I'm just gonna click this auto hide push pin, which puts it over to the side. If I want to look at it, I can just put my cursor over this little library match tab and it will slide out, but it's not going to be a point of real interest here. Now, immediately after importing some data for the first time, what I want to focus on is the quality of the data. So let's bring up some other views. So view, the replicate comparison plots are almost always a must. Let's bring up the peak area replicate comparison and the retention time replicate comparison, and I'm going to drag these over and dock them on the right side of my window here. So I do that by clicking as if I'm just going to move a window around, but with these are dockable windows, and I get a big blue box, and then I can bring the cursor into one of these docking indicators. And once I get it there, the blue box shows me it's going to be attached to the main window in this position. So I click that, let go of that. And then I'm going to take peak areas and drag it similarly, and drag the cursor into an area that tells Skyline I want it to share the same region as the retention times. So there they are. You know, if I wanted to give them more room, I could actually have them share sideways, which can be sometimes pretty, with this few replicates, that can be pretty nice. And we get more of a sense of like how consistent is the retention time of these things. You know, we want to see pretty high consistency, and when we spread things out this much, maybe we can see just over the range of a couple minutes there is some drift. So they're getting, I guess, later over time. But if we look at this as a whole and click on the IRT protein, then now we can see it's pretty stable. They are pretty stable, but maybe notice over here that the peak areas are going up and down and up and down. And with I generally try to teach people in the courses to, you know, make sure that you have a clear idea in your head of what you're expecting, and then look at these plots and say, do they match what I'm expecting? So we're expecting, ideally, what we'd like to see is retention time totally flat across here. And I can actually switch these to be a line graph by right-clicking and making the value, rather than showing everything, just show me the retention time. And now I get a line. And now I can have a real good sense of like, okay, yep, there's some drift in the chromatography slightly later, but it's really pretty stable. Up here, there's, you know, more up and down than I would hope. And again, these are peptides. The IRT peptides are all peptides that were injected in a constant concentration, so I should expect a line straight across. And then when I pay a little bit more attention to what this up and down pattern is, you can see that it's down in A and up in B, and down in A and up in B, and down in A and up in B. So if that pattern it is present in the rest of my data, then that means that I've got a bias in my data, right? Because I'm intending to do a group comparison between A and B, and if things that are at a constant concentration are going up and down, based on whether they're in A or B, then there's going to be a bias in this data. I can quickly check that by looking at a human peptide or a human protein, rather, and it's not as clear. But maybe here we see the same sawtooth pattern a little bit. So we'll see. But yeah, so there is some evidence of sawtooth, and these also, the humans are supposed to be at constant concentration. So just something to keep in mind. This is just sort of a quick initial analysis. Another thing I might want to do is I'm going to shift back out to Shift F11 to show the entire chromatogram graph, and then I get what is called, what Paolo Piccati called the protein fingerprint. Every time I select a protein, I can see all of its peptides. And I'm going to go to view arrange graphs, and I could just do tiled, and that would give me 6 plots like this. And maybe if I gave them a bit more room, that's actually a pretty good view. Or I could go to view arrange graphs grouped and say I want 2 groups, and I'm going to have them in a row and document order, and I want to distribute the graphs among the groups, which it tells me, if I read it, it says like dealing a deck of cards, and that will put all of the A's on the left and all of the B's on the right. So maybe that's a useful view for me. So now some more data, before we get into the goal of this is to do a group comparison, which we're going to have to get to fairly quickly because time is going quickly here. But before we do that, I'm going to show you a few more ways that we can get a sense of the data quality before we do our inferential statistics, which is always a good idea. You may just by looking at some of these plots, you may realize, oh, I need to re-import this data. It's just not, or there's some problems and I need to kick out some things. So the first thing we'll look at is, well, let's take another look at the retention times by looking at a regression plot. So we're going to do a regression of score to run, of those IRT scores. So putting it right here, but I'm going to drag it out, make it a little bit bigger. And so here you can see, well, this is, really nice correlation. If there's a slope and an intercept, now the measured time is on the y-axis, and so the independent variable is the score. So those are our trained scores, and then the dependent variable is the retention time, because I'm trying to build something that will predict retention time. So I can choose what points to plot. Right now I'm just doing all of the targets, and I could do targets at 1% FDR. That gets rid of some things. It's an even nicer, better correlation here. And I could also you look at the decoys. How do they compare? And you can see, wow, they're really quite different. And so that gives you some insight into why retention time is a good score in that M-profit model. A better way to look at these even is to, or another way to look at them that can be very informative is to plot the residuals. So that just shows us how far away from the predicted retention time. And there are some things here, some of the decoys, we just didn't even have any signal that we could call a peak. So these are basically on the x-axis. But if I zoom in here, I can see, okay, the decoys are spread, again, remember I limited everything to plus or minus 5. So they kind of spread out to plus or minus 5, but they cluster closer to the center, which is maybe just, the way the probabilities work. But now we want to take a look at what do the targets look like. So if I go to points, targets at 1% FDR, then wow, you can see really a lot better that, again, as I said, I could have limited my extraction to 2 minutes, plus or minus 2 minutes, and that would have been just fine for this data set. We can zoom in here. If I put the cursor to the left axis, then that allows me to do mouse-centered zooming only on that axis. And you can see that it's not exactly a straight line, that there is, this curvature going on, but a straight line still does a great job of approximating, there's really not much that's more than, half a minute away from where we expected it. That's the vast majority. And, it's quite possible that these other things that are further away are just our false discoveries. But it can be very useful to look at it this way. Sometimes you find out that there's some really bad curvature that's going on. There's, there can be problems in the chromatography that you can see here. No problems here. You might also just say, yep, next time I run this, I'm going to do plus or minus 2 minutes. So that we've learned something from looking at this view. I'm going to close it. The next view I'd like to bring up is the mass error view. So usually good idea to have a look at the mass errors, especially since we use them as Ben explained, we use just in a mass extraction window. And so if the mass errors don't match our mass extraction window, we can end up with pretty bad results. Here you can see that, in general, we just see almost nothing out at 20 ppm. So, that's a decent window. And if we went plus or minus 10 ppm, you can see that we might be cutting some things off. This is a single replicate. I can also say all replicates. So if I'm doing a single replicate, I can actually look at each individual replicate by clicking on the tabs or choosing stuff in this replicates drop down here. So you can see that there's some shifting around, and actually there are some replicates that were 5 ppm off, and so there is some movement. And I guess, yeah, the later replicates maybe, well, it's not even the later replicates. I'm not quite sure. There's maybe a pattern that can be worked out to which ones were shifted. And then if we went to replicates all, this would give us something like this. And you could see if we had a 10 ppm cutoff, we would be losing some of our data. So that might be an exercise for future. And this is an interesting way to look at things. Another interesting way before we leave mass errors is actually I can right click and go to the 2D histogram, and I can have a look at whether or not there's any correlation between retention time and mass error. You might see, oh, wow, this is curving upward, but here it's pretty stable. And we see again, that mass error sort of center value is not right around 0, but it changes by each replicate. So it's almost always a good idea to at least look at this single replicate view and see how that's changing. And you can see that maybe in this one there's a little bit of drift in mass error over time, and then I can also switch to mass to charge ratio as the x-axis, and now you can have a look at, maybe are the low mass to charge ratio ions having a mass error problem, or whether or not there's any sort of curvature in this plot or drift in this plot, and there really isn't in these data. So enough with mass errors. Moving on to last thing we're going to check before we get into the actual group comparison. As a plot, mass errors were actually added by my son Alex, and now we're going to go to peak area CV histogram, which was added by Toby Rode, one of his best friends from high school, who's now at the UW, and has done a lot of great work for us. So here are the CVs. And at the courses, I like to ask, what do you expect of CVs in a quantitative experiment? And usually there's somebody who can say it's got to be under 20%. And so here we have below 20%, and you can see, wow, it's 48% are below 20%. And then, the real puzzler is, what are these lumps out here that are way outside the 20%? I see 3 distributions. And so the important thing to note here is that this is across all replicates. We've got 6 replicates, and we actually are expecting change. So when you're looking at CVs, you generally want to look at things where you're not expecting change. And so these lumps represent the other organisms. So this is definitely the human lump, and then this would be yeast, and the one that's the farthest out is E. coli. So we want to try to get to a histogram plot that gives us a representation of technical variance. And in this experiment, actually, the A's and the B's are technical replicates. They're not actually. Not every replicate is its own biological sample. So before we do that, we need to, before we can do this, Skyline has this ability to group by an annotation, but we need to annotate these replicates. So I'm going to go to the document grid, and I'm going to switch to the replicates view. And what I need is you can see these are some calibration values that are there by default. And here are my 6 replicate names, but I need an annotation that allows me to tell Skyline about the sample mixes that I have. So I'm going to go to settings, document settings, and it comes right up with annotations. This is a brand new pristine installation, so it has no annotations. I'm going to add an annotation called sample mix. Whoops, that's sample max. And then I'm going to make that a value list, which has potential values of A and B. And then I tell Skyline this applies to replicates. So you can annotate many different objects in Skyline. It's already checked, so that's all I need. And then you can see that column shows up here now, and I can just type in it A, B, A, B, A, B, and I'm just matching the A's and the B's to what I see in the replicate name. And I highly recommend you get used to this document grid. It's your key to getting reports out of Skyline, as well as, and they're highly customizable reports. And it's also an extremely powerful, flexible way of accessing all of the data values that get plotted in here. So, but we're going to leave a deep dive into the document grid for a different Skyline tutorial because we're already running late here. So now I can right click and say group by that sample mix annotation I just added. And okay, so now those lumps are gone. I actually have a single distribution. Yeah, I could look at each one of these individually, A and B, and A looks like it's got a little bit more variance than B. But I go back to all, and then now I can choose to normalize to something. In this case, I'm going to choose to normalize to medians. That's not always the right decision. In a targeted experiment, you don't want to normalize to medians because you can't necessarily say that you have the expectation that most of the things in this sample are not changing. Luckily, with all of the human proteins not changing, we can still say that we don't expect change among our median values. And so we normalize to median, and this is actually something that Ben pointed me to when we were working with his pan-human library. And yeah, median normalization works great here. Now we have 85% below, below 20%, and the median CV is 6.5%. So really quite high quality data here. So that should give us some confidence. I'm going to close this, and we're going to end by creating a group comparison. And so we can go back to document settings, go to the group comparison tab, and add a group comparison. And we'll just call it A, V, B. So A compared with B, and the control group annotation. The annotation we're interested in is this sample mix. Control group A, compare against B. We don't have an identity annotation here. That would be if we had technical replicates of subjects, which we don't have. We can say we want this equalized medians normalization method. We can specify a confidence level, 95% confidence. This is unadjusted, so it's not going to be as good as using an adjusted p-value, but it gives us it could give us some insight into the confidence we have in our means. And then I'm going to go ahead, I'm going to apply to proteins. We're doing protein comparison here, which is going to just give us fewer points and then more values to contribute to each measurement. And then we're going to use sum of transitions because we have an m-profit value, an m-profit model, and we've assigned Q values. I'm going to set a Q value cutoff of 0.01, so 1% false discovery rate. And what I'm saying is that if the peak, so we have these peaks, these detected peaks that we match to peptides, if the peak is not within 1% FDR, then I don't want to include it in my quantitative measurements. So let's go ahead and say okay to that. We have our group comparison, close, and now I can view the group comparison by going down here to other grids, group comparisons, and A versus B. So there it is. You can see I've got 2,187 proteins being compared. So I've lost some proteins, and those are ones where my choice to exclude certain peaks gave us too little data to make a comparison. And so I did lose some things, but I have quite a fair number of them. I could I can sort and filter in this view here. I can move that out. You can see those are the human. What I'm going to do, and so yeah, you can see that the human fold change results are really quite close to 1 mostly. And then if I come down here to yeast, I can see a lot of 2s. And if I get all the way down to E. coli, I can see hopefully things that are close to 0.25 or 4-fold. 1 and 4-fold change. But a better way, now we'd like to plot that, and the best way to plot it is this volcano plot, also created by Toby Rode. And here is our first volcano plot, and you can already see that there's a bunch of stuff that lines up on this essentially 2-fold. Log 1 of log 2 is 2-fold, and then -2 of log 2 is 1 and 4, which is what we're expecting for the E. coli. And then there's some stuff in here which is probably human. Actually, you can see that Skyline will highlight what I have selected. So as I move the selection around, you can see that Skyline's showing me the point, and I can click on things where, yep, I expected that to be yeast, and it is yeast, and when I click on it, then I get all the graphs, and so I can navigate around my data, and this over here I'm expecting to be E. coli. If I hover over it, I get a bunch of statistics about it already, but if I click on it, then I get selection, and I can see the graphs for it. So, but it would be easier to understand if we could actually color things the way they're colored in those plots from the Navarro paper. So I had Toby implement that as well. And so you right click here and say formatting, and we first need to assign an expression. And we're going to do the expression based on the protein name, and you can see that all the protein names end with humans for humans. So we just say human here, and now you can see it's all human. So that's a pretty simple regular expression. You can get lots more complex if you know enough about regular expressions, but that's all we need for here. We're going to assign a color, and the color of human was green, and click OK, and then do the same for yeast, which we just say yeast because it's actually yeast 8. I don't really understand that, but click OK, and then that color was sort of an orangey color. And you can see already that Skyline is highlighting the graph in that way. And then finally, boy, I hope I got these colors right. Maybe not. And then we're gonna, again, on protein name, E. coli, and then give that the color purple. So, and then okay with that. And now, so here it is. You can see these are the, these are what we're expecting. Anything you do over here gets reflected over in the plots. So for instance, I can actually limit myself. So if I click on the header, I can sort, and I can filter. And so I could limit my filter to contains human. And so there are the human things, and you can see maybe, well, maybe there are a few things that are over here, and maybe there are a few things that are, this is So this is something, maybe these 2 points are things that were actually E. coli that got identified as human, and maybe these 2 points over here are actually yeast that got identified as human. You can also see, I think, that slight bias that we talked about. So you can see that the human, rather than being really centered on 0, which is what we would expect, you can see that they're slightly off to the right of 0, and actually we get some really highly significant changes. So this is super significant change of some pretty, let's go even closer to 0, 1.831. You know, that is considered a very significant change of a very small amount. And so that just means that by some lucky chance, we got very consistent measurements between A and B, and our statistics say, well, that's, you know, certainly that's a real change because it's got a p-value, a q-value that's really high. So that shows you how statistics can sometimes be wrong, and why people sometimes choose a cutoff that, okay, I'm just not interested in things beyond a certain range. Let's quickly just all switch the filter to E. coli. And, you know, maybe this is actually, certainly there's some things that aren't changing, but that could be, you know, that could just be, you know, the really low things that are below the lower limit of quantification. So that as their signal changes, they're not actually changing. But over here, maybe this is actually, you know, or these two are actually yeast peptides that got identified as E. coli. And so now I'm going to end that walkthrough. We're running late here, got to get to your questions. All right, it's Brendan back here. I'm going to get rid of this. So I'm going to, and we're going to continue with our session. Hopefully people found that that webinar useful. And I'm gonna, so yeah, we've done this. We're gonna now actually take your questions rather than the questions that were asked at the webinar. I've been answering some just in text as we go along here. And I've saved a few to answer in general. I do wanna also point out, I'm gonna, again, share the results of the poll, and you can see that, you know, a lot of people said that they're either pretty confused or they think they understand. They've been watching taught lectures and reading papers, and they think they understand DIA. To get to this level where you can do your own experiments or you're using it regularly, you really need to sit down and do these tutorials. So if we were actually at the May Institute in Boston, we would set aside some time and make you do the tutorials. But the nice thing is we'd also be there to help you. So you could just like raise your hand and go, oh, you know, like I'm stuck at this point in the tutorial. So what I think I'm going to do is when this session ends, I'm just going to stay online for a while if Mina and Olga will allow me, and then anybody who wants to start the tutorial after this session can go get the written tutorial, start and ask questions, and I'll try to stick around for an hour. But anyway, so let's yeah, so that's the I'm gonna stop sharing these results. And then yeah, what we also found from these polls was that this is, most of you didn't actually do this. So you're hopefully you can set aside some time today to go back and do the tutorial, either proteome wide or the simple tutorial that we usually have people do is just a selected set of peptides. All right, I'm also going, I'm going to get rid of this PowerPoint slide. I'm going to now switch. That's going to be complicated. Okay, well, anyway, I'm not going to quite do that yet. In a moment, I'll switch to my other computer, which is the computer I demo on, but that's going to take away the questions. So let me see if I can answer some of your questions. There are quite a few. So here's one question that says, which software do you recommend to use for DDA analysis to create spectral library from your experience? And I would just direct you back to this Skyline website, and the library tool is called BiblioSpec, and you can then get a sense of all of the BiblioSpec's support, and especially this file formats here shows you all of the different things, all the different search engines we support, and it's quite a lot. So you get your choice. There's a nice paper by Olga Schubert. And they graciously added me to this paper, but building high quality assay libraries for targeted proteomics is a great paper. It takes you through all the Aebersold lab best practices for building these, and I would say that they have built some really nice spectral libraries. Ben Collins's own pan-human library is kind of set the standard for human libraries that can be distributed. This tutorial takes you through doing it with Spectrast, which so you don't necessarily need to do that, but certainly all of the steps up to producing the data that you build a library from, and then I would recommend that you build the library in Skyline as was shown here. And then, so I'm going to count that as answered. Lots of different tools. I don't have a specific tool, Mascot, MaxQuant, there's 20 different search pipelines that work great with Skyline. So it's really more how you produce the data. And then the next one is the asking about the paper, the DI swath paper, and that Ben mentions, and that is 2018, yeah, so that must be this one. It's by Tina Ludwig, and includes Ben Collins and Rudy Aebersold, and this is a nice tutorial paper that goes through a lot of useful information about DIA. It's an attempt to sort of apply the same tutorial and instructional format as the Longa paper, which also came out of the Aebersold lab for SRM, and that's a really nice paper. So if we do not have IRT peptides, so if you didn't spike in some IRT standard mix, you saw that set of mixes. We chose Biognosis, which was the first set, but there's lots now. And if you didn't do that, what should we do for building the library? And you should use endogenous peptides, and Skyline makes that easy now. We're working on that, and I'm going to show that tomorrow a little bit. So there is a way to use endogenous peptides. There's a really nice set of conserved peptides, which were described in this paper, Parker, Aebersold, Roast. So this is a paper by Sarah Parker from Cedars-Sinai when she was visiting the Aebersold lab, and they came up with this this set of peptides conserved in eukaryotes that we use as a basis for choosing IRT peptides. But even if you don't have those, we can generally come up with others, and Kaipo Tamura has done a really nice job in improving that recently. So we'll talk more about that tomorrow if you show up. Somebody is asking about what does the term centroid mean, and I guess so. What centroid means, is that I'm going to go to the Skyline tutorials. I've recently redone some of these, and this page, you can, you can almost see that is not a centroid. That's a profile peak. There's a distribution. This is a mass to charge ratio. This is an isotope distribution in a mass spectrum. And what centroiding means is that each profile peak like this just gets turned into a single stick, and we go from thinking about extracting. So this yellow is the extraction range. We go from thinking about extracting the peak by a resolution to extracting the peak based on its mass error, because the peak is now just a single spike. So that's what centroid means in this context. What would be your threshold for dot product, dot P, when you accept the correct identification? So dot P is so Skyline actually has 3 sort of dot P values. One is the I dot P, which is the match between the isotope distribution and the expected isotope distribution. So it'll take these peaks and say, well, we expected them to be distributed in a certain way. That's usually pretty good. And in that case, sort of below 0.9 is questionable. You can even see it up here. This is maybe a little too small to see, but in this case, it says the I dot P is 0.99. So in these data that I'm showing here, I have extracted precursors. In the tutorial I just showed you, we didn't extract precursors. But in this case, the fact that I have a 0.99 isotope distribution, a very nice peak that coalutes with my fragments, gives me extra confidence that I've found what I was looking for. And then you can see that the dot P here is 0.83. And I would say, for me, if you have 6, this is, I guess, 5 transitions, you have 5, 6 transitions, a dot P above 0.8 is pretty good, and I would be highly suspicious of anything below 0.75. But again, we don't tend to use these as cutoffs or thresholds when we're using M-profit. Then we use the statistics of the M-profit model, and we're looking at Q values. So, but just as a general thing, those are the available dot P's. The third available dot P is the ratio dot product, which only applies to when you have an isotope labeled standard added. So done with that one. Could you later explain a bit the peak scoring model and different components? And I'll do that in a second when I actually switch back to where I can have Skyline up and we can look at the data that were just processed. I'm going to switch to the machine on which I processed it. Right now I'm running off of a different machine, so I'll get back to that one later. And then does refine reintegrate give same effect as filtering by M-profit Q value? Yes, refine reintegrate is a way to apply an M-profit model and get Q values. So I think, yes, refine reintegrate is your method to getting to M-profit and the Q values. So let's see, if we select TMP for protein summarization, does it give same result as MS Stats protein summarization? And I say no. You know, that Tukey median polish, TMP is Tukey's median polish. We don't claim to be the same as MS Stats. MS Stats is a lot more sophisticated statistical tool created by statisticians. We created our group comparison working with Mina very closely and Olga very closely. But if you want, I mean, there's a paper coming out from Mina. I've seen it. I think it's maybe in revision, hopefully be resubmitted soon, showing how MS Stats does quite well. And so, yeah, later we're going to, so you know, think of Skyline as sort of a rough cut. If your data quality is really high, Skyline frequently will be all you need. But if you want to do more sophisticated statistics, then we're going to point you to MS Stats, and we'll get into that. You know, tomorrow I'm going to show you how to go through Skyline and create a report that then can go into MS Stats. And then next week, Mina Choi herself is going to be describing how to use MS Stats and make the most out of that. Do you recommend using both target decoy and second best peak for building M-profit model? I never have a second, so especially in DIA, I think the standard is to use decoys. In cases like PRM, where you haven't actually, in targeted cases, to get decoys, you need to think about that ahead and target things that are decoys. And so in cases like PRM, more frequently somebody will use second best, and we don't tend to mix the two. Second best to me is second best. it's something you use when you when you can't have decoys for some reason, like you already targeted your data. One thing I would recommend is, if you're using second best, I would recommend not using retention time as a score, because by definition, if you're using the second best peak, you're eliminating a retention time space where the best peak is. And so the second best peaks will almost always be further away from the predicted retention time than the best peak. So something to think about. Whoops. I have a lot of history with Spectrast. Where does it fit in these days? So you could still use it. Skyline totally understands Spectrast. I just think it's more cumbersome, more work to get a Spectrast library. My own feeling, as I've become better and better, you know, or I've made Skyline's code better and better at building libraries, is that the DIA libraries, there's, especially with retention time IRTs, there's some tricks to it that I'm not sure are reflected in Spectrast, and I'm not sure it's like actively under development now that the Aebersold lab has closed down. So you know, there's still instructions for how to use it from the Aebersold lab. You still certainly can use it, and Skyline will understand it, but I will, you know, it may be sort of aging as we speak. Whose centroiding algorithm is used? So yeah, we always use the vendor centroiding algorithm. So yeah, Scott, there, you know, we're underneath the covers, we're using the ProteoWizard data access layer, but Skyline never relies on ProteoWizard centroiding. It's always vendor centroiding. And so yeah, if you have a problem with the centroiding, talk to the vendor. Have you compared the results of MS1 filtering versus DIA, assuming you run the same number of injections, et cetera, et cetera? Yes, I mean, and we've compared. Yes, there's been lots of comparisons between DIA and MS1, and even in this screenshot over here. You know, I've got both the MS1 and the fragment ions, and when I've done exactly the same integration, I now have lots of large data sets where I've been able to compare what would the quantification be if I use the MS1 versus using the fragment ions. And it's basically lower precision. I mean, you just can expect MS1 is less selective, more noise. There are problems with dynamic range because the MS1s come from a survey scan, and so instruments like the Q Exactive, you know, have a certain fill time. They inject a million ions into the Orbitrap, and if you have a low abundance ion, it may not be captured well. And people have been working on methods like the boxcar method to deal with this. But in general, we feel pretty confident that MS1 filtering is not as high precision as DIA. When generating spectral libraries, what filtering do you recommend if there are in DDA search engine software prior to Skyline? Yeah, so I would recommend trying to get to a fairly low false discovery rate, and then you have to think about your search engine, whether or not it just has a false discovery rate or whether or not it has some other probability like a posterior error probability, and you have to convert to false discovery rate. And of course, this will be different based on whether or not you're doing fractionation and lots of runs, which will start to try to inflate your false discovery rate. And I think that I hope that article from Olga Schubert that I mentioned at first, Building High Quality Libraries, treats that subject, and you should have a look there. What language was the Skyline user interface written in? Skyline is written in C#. All right, so I'm gonna move on to now. We're gonna switch to that computer that I ran everything on. Here we are. This is the folder. This is basically if I drill in here, this is the document I built that you saw in the webinar. And if I double click on it, I should be able to bring it up in Skyline, and we will be right where we left off. So this is where I left things. Here you can see at the top the IRTs, and this is one of the human peptides. I've got things normalized right now. I've got things normalized to total. So this is a view in Skyline that shows not the actual peak areas, but just how the fragment intensities, how constant they are, and we expect them to be very constant. So you can go through here and see like, okay, yep, these are very constant. And you can also see this is the expected library intensity, which doesn't match perfectly. The library intensity seems to have a high amount of B2, which is not showing up here. Actually, yeah, that's okay. So they don't match perfectly necessarily. One is DDA and one is and the other is DIA. So but across the runs, they're very consistent. I can turn that off by going over here and saying normalize to, and you can't see the other menu because it's popping onto my other screen. But if I just normalize to none, then now here I can see the actual peak areas, and I can see the .p values that tell me how well it's matching this library. All right, so somebody asked about, so one of the last questions in the Q&A was about this refine, reintegrate, and let's look at the scoring model a little more closely. Kind of went past it very quickly. So this is the scoring model that got built. And if I just do edit current, I can open it up. And there it is. It's a really nice scoring model when you get this kind of separation. One thing to note is that there's really very little under the decoys, meaning there's really very few false discoveries here, as the statistics analyze them based on this distribution. And that's because we started with a sample specific library with no fractionation. So this is what our targets are just things that we already measured in DDA. So we're not trying to build a deep fractionation library, which is what we're going to look at tomorrow. Here is the model. Anything that's highlighted in red and has a negative means it's going sort of the opposite direction of what's expected. And I would say when you get these, you might really seriously want to think about unchecking them, because this means that the score is being weighted opposite of sort of what is physically expected of this. So it would be red if there was more intensity in the decoys. That's, that might be informative, but it's not necessarily real. Like, okay, so yeah, decoys somehow get more interference, and that's why their intensity is higher. But like, is that really a good thing for you to be picking your peaks on? And then retention time difference. We used to have retention time difference squared here, and that frequently would end up negative, and then that could have the impact of things that were really far away suddenly got great scores, so that you have to think about, does it make sense for this weight to be positive? The further away you are from where I predict, the better. That doesn't make any sense, and so if that's what your scoring model is saying, you might want to think twice about including it. And so, but everything here, for the most part, there's just this one that's red. It's very, it's very low percentage. I wouldn't bother unchecking it in this case, but if I wanted to, I could just uncheck it and retrain the model. And Skyline could do that pretty quickly. Most of the scores are described, and so that really hasn't changed the model very much. Nothing else has gone red, and the sort of relative distributions are still very similar. Signal to noise is almost a useless score, but most of the other scores are adding some value, and it's really nice to see retention time difference, which can be, sometimes it's really strong, and if your IRT measurements are not super strong, and you haven't, and it's not wide enough, that might not be a great score. We can take a look at the actual feature score distributions by clicking this tab. And so now you can see, well, this is what shape looked like. And to me, it's a pretty good, it's actually a pretty good, maybe not a perfect normal distribution, but it's a pretty good discriminator, but it may have very similar discriminative power to some other things. So that sometimes you'll get a negative score just because, well, this score separates things very, very similar to another score. We can look at, here, this is very nice and normal, and we see more intensity on our targets than our decoys, so that's a good one. This is not exactly normal distributions, but and maybe our buckets are too small, but you can see that way over here at the very low end, there lots more targets have are very close to the are very close to the predicted retention time. This is the dot product score, always a really good score. It's very normal. That is which is what we're expecting for the linear discriminant analysis that's used by mProfit to build these models. So that's a really nice discriminative score. If this is not a good score, then there's something wrong with your library and you should go back and reassess what you're doing. That's coelution. Also, yes, there's more, coelution, obviously more targets are close to having 0, but, and then there's this thing coelution count, which this was Skyline's original score, and you can see it separates things pretty nicely. And, but it, and it usually has a pretty high weight as well. It's just very sort of, not very granular. Signal to noise, okay, yeah, there's some shift, but maybe it's very similar to other shift. And then product mass error, maybe just adds another dimension. So those are the components. These are the p-values, which are just generated from this model, just taking each score and saying what's the probability of being under this orange distribution. And then there's a method called Story Tibshirani, published by Story and Tibshirani, that allows us to come up with the Q values. And so that's how you can look at things. I highly encourage you to get a sense of this and to know when things are wrong. One thing that we look for is none of these scores should be grayed out. It could be possible that if you didn't get your retention time predictor right, you would have a gray retention time difference. And you shouldn't just continue on and say, oh, well, forget it. Retention time difference is an important score. You should figure out how to fix that. All right, so we're already to 2 hours, but Holga has told me I can stay as long as I want, sort of. So I'm going to move from questions to what I promised the Prosit people. There was a at least a quarter of you that were only here for Prosit. So let's start to think about Prosit. When we start to think about PROSIT, we're going to want to start looking at this library match view again. Let me see, I'm going to put it down here and expand a bit so that we can see it better. And so the first thing to do to work with PROSIT is to go to Tools, Options, and PROSIT. And here it says server unavailable. I kind of want to change that message because the server is totally available. It's just that we haven't chosen a model, and there's actually just one model for intensity and one model for and one model for IRT, and it just, and now it says, hey, server's online. So as long as we choose the models, then Skyline goes out and checks those endpoints and says, yep, they're there, they're ready. And you know, definitely when we were testing, we probably had 3 of these intensity models, and we've always had 1 IRT model. So that's enabling PROSIT. Once I've done that, I guess the most interesting thing is going to be looking at this spectrum plot to start, and I can just I could just go like this, and now I can see the PROSIT predictions. So you can see these are completely clean, and as I change my selection, Skyline talks to the PROSIT server and gets its predictions. And then the next thing that would be interesting is to look at this mirror plot, because that tells us how well it's matching what we have in our library. And so you can see the noisy spectra at the top and how well that the things are matching, and you can see 0.7. So it has been, this is the dot product between this upper spectrum and this lower spectrum, and it has been suggested that we not do a full dot product, which is including all of these noise peaks. So in a cleaner spectrum like this, we get up to 0.83, but if we just if we got rid of some of, if we got rid of noise peaks like this, then we would have even better dot Ps. But you can see that these are a lot of ions, and mostly they're above 0.5. When you're matching this many ions above 0.5 in a spectrum, above 0.5 is quite good, and you can see that they really match quite nicely. So that's a first way to start getting some understanding of PROSIT just at a just at a very descriptive level. But what we're interested in today is creating a new library. So I'm just gonna I guess I'll leave that up. So we would like to create a new library. So we've got our spectral library that was collected by Ben on his own data, and we're going to create a completely new library, which is going to sort of batch request spectra for all of these peptides as well as IRT values. And so I'm going to go to here. Well, let's save this file, save as, and we're going to create a new document. And what we're going to try to do is replace our replace our current library with our experimental library with a completely PROSIT predicted library. I don't understand. Oh, it's so it's updating library settings. So there we go. Okay, now fully saved and ready to go again, but we're going to go replace the library. So we go to settings, peptide settings, and library, and we've got this. Oh, it's given us this new thing. But that's not really a PROSIT library. So we're going to click build, and we'll go. So we got this, we're going to create this new PROSIT predictions library. We're going to use PROSIT, and then we want to tell it, again, here we can choose whatever standards we want. We can add to this standards list. You can see all the like Pierce, Replical, SciEx has its own. And then the CIRT one is from that paper from Sarah Parker. But we're gonna, we want to stay with our diagnosis 11. NCE 27 turns out to be just fine. I've actually tried this with these types of data. This refers to a thermoparameter, normalized collision energy, and that's because the predictions are done with this. And 27 turns out to be a good sort of average value, and it works fine with triple top data. I've tried it. So we'll click finish, and then that's, oh, I got to tell it where to put that thing. And so this, we'll put it right here. And finish. So you can see the progress it's building right here. I'm just waiting for it to finish building. And then, there it is. It's got, a nice regression with so the predictions match. They don't match perfectly, but like we can do a regression. So you can see that this is not, this IRT value is negative 20 something, and it's not quite so there's a shift going on. Yeah, so this goes all the way out to 140, and this is up to 100. So PROSIT is not giving us exactly the same IRT values, but we can recalibrate everything back into the space that we want. So, and Skyline saying, yep, that's going to work great. There's a slope of this and an intercept of -20. So there's basically PROSIT is shifting things more or less about 20 over in its predictions. So then it says, do you want to recalibrate the IRT to your current data? And we'll say no in this case. Last time we said yes. And then it says, do you want to create a retention time predictor with a 10-minute window? So that's going to switch. It's going to use the IRT values in our library from the IRT values in the old library. And so we'll say yes about that. And so now we have a PROSIT library. That's the one we're using. And it's got to take a moment to look at it. It's actually repicking the transitions as well. And now you can see, oh, hey, dot product of 1.0 with your library and the PROSIT prediction. That's going to be the case in everything. Everything's going to be 1.0 because our library now contains the PROSIT predictions. So there's no difference here. And now what I'm going to go do is edit, manage results, and I'm going to remove all the results. Actually, maybe we can, yeah, so you can see that Skyline or that PROSIT doesn't, isn't exactly matching. So where you can see an empty dot there, that means that PROSIT has a different opinion of what is this is rank 5, and the old library had a different ion. So you can see that they agree quite a lot, but sometimes they don't. And it's usually like rank 6. It's usually at a very low rank that they're not in agreement. So yeah, PROSIT prediction's doing pretty well. I haven't seen anything where I'm getting well, here's something where it's disagreed at 2, 4, 5. So they basically seem to agree on the top 3 ions, and then there's some disagreement on the lower end. But you can see that Skyline is now rechosen all of the transitions based on what PROSIT, how PROSIT's rankings are. So now I go to edit, manage results, remove all, okay. And before actually, I'm going to undo that. I don't need to remove them. I'm just going to re-import them. But before I do that, I know from my own work and a lot of testing that the PROSIT IRT predictions are not quite as good as empirical ones. amazingly, the spectrum predictions actually can be sort of produce indistinguishable results to even slightly better results, which we sort of attribute to there being a canonical spectrum and your actual measured spectra having its own variance. So it's a little bit like using an isotope distribution where we can calculate ahead of time what the distribution should be versus having the variance of the instrument involved. So let's go to the transition settings, and here I would recommend with PROSIT that rather than having a 10 plus or minus 10 window, we're going to, or plus or minus 5 chromatogram window, we're going to go with plus or minus 10. And that's pretty much the only change I would make here. And then I can go to edit, manage results, and I just shift and select and re-import, so that puts a little asterisk by them, and it's gonna re-import everything. So click OK. All right, and now we're gonna wait for it to re-import, which should take close to 2 minutes, I think, maybe a little longer because we've done longer chromatograms. First, it does the IRTs, and then it comes back and does the extraction of everything else. So I'm going to switch back to questions. There are a few questions. So let's see, what do we have? Where do you access this scoring window? Oh, well, so somebody's asking about the scoring window, and that again was refine, reintegrate. And we're going to redo that in a second. So I'll show you in a second, because we're gonna do a refine, reintegrate on this data, and I'll show that. What is the purpose of PROSIT? It's a predictor. So this is a neural network that's running out on a server. It's a server in Munich, and or not Munich, Freising, Germany, at the Küster Lab at TUM. I visited there last year, and I'm feeling a little sad that I may not make it to Europe this summer. But yeah, it's their server. It's running on GPUs. It's not something that we could easily integrate into Skyline running live. So we talked to a server to get these predictions. And as you saw, it predicts spectra without any noise. So they're not, but you can make libraries out of them. And then it also predicts retention time. And they're adding new predictors, which we will definitely integrate as well. One for CCS, collisional cross section for ion mobility. One of the things I'm sort of cheating on here a little bit is I'm still using the peptides and precursors that were found with my DDA library. So the Küster Lab is working on something that will predict what are the best peptides and precursors to include. So right now I'm using empirical data to choose my targets, and then I use the library to choose the transitions. And in the future we'll use PROSIT to choose everything. But what we have found is if you just exhaustively use every tryptic peptide and every charge 2, 3, and 4, you can do this. You can ask PROSIT to predict these spectra, but the false discovery rates will be inflated. So that's PROSIT. That's what PROSIT is a predictor of a number of things, useful chemical physical chemical properties of peptides. So yes, and so that got sort of answered again, or asked again. Do you have to have IRT spiked into samples for building a PROSIT library? So no, you don't have to have. You can build a PROSIT library without any IRTs, and you can use. Yeah, let's see. If you wanted IRTs, yeah, I mean, you can choose your IRT standards to be anything. And so as we saw with, so settings, peptide settings, when I did the build, and I went with PROSIT, I get to choose the IRT standards. You can even add them now. So you can add your own set of IRT standards. You can have spike-ins. This CIRT is, those are endogenous. So no, you don't have to have any spiked in peptides to do what I'm doing. So PROSIT is something you cannot have access to. I think that's just referring to that you have to turn it on. So yes, you can decide you don't want to have PROSIT turned on, and we can now go back here and we could turn it off like that, and then Skyline will behave in a certain way. But if we leave it on, then we get access to PROSIT. So thanks to the generosity of the Kuster Lab for having the server that responds to our requests. How good is PROSIT with post-translational modifications compared to your spectral library, say phosphorylation? So I would say go back and, well, I think they're working on those things. They definitely support methionine oxidation. They support a restricted set of modifications, and I guess you would have to see if they support yours. I'm sure they're working on phosphorylation because that's so interesting to everybody. But yes, they don't support all. They don't support the complete set of post-translational modifications that Skyline supports. So we've done the import, and then the next thing to do. So that's re-imported, and you can see now everything has a dot next to it. And I'm going to close the spectrum match window, and we would now go back to refine, reintegrate, and we're going to not use this model, but create a new model. And Skyline does some analysis. Ooh, I don't know why that's happening. So this is an example of what I said, the retention time difference could, I know why it's happening, darn. Maybe, I'm not totally sure why it's happening, but let's see what I'm gonna, so this is where, there's no retention time difference. We can actually go and view, so we want all of those scores to be enabled. That one's grade. We might go and look at the regression. Score to run. And here are a bunch of things that are, so let's see, so we're looking at the DI-TTOF library, and we want to be looking at this one. So this is the library we created originally. This is the library. So you can see that there's a bunch of things that are early eluders that are chosen very differently with PROSIT than without PROSIT. So we can switch to our PROSIT predictions, and now everything should be is within the range of what PROSIT allows. We can also switch to that the residuals plot that I showed in the tutorial, and you can see, so some things, there's actually most things in this case are being found within plus or minus 5 minutes. But it's certainly, there's some curvature. It's certainly not as tight as we saw with the old data for retention time prediction. And usually you don't want to just like cut off really close to your actual distribution, because then the retention time won't be a good score. So now that's looking at the targets. And then we could look at the decoys as well. And that should be a more, bigger distribution. And if it doesn't work, maybe there's something wrong with the decoys. So one thing I forgot to do, and one thing we're going to improve, is that I forgot to regenerate the decoys. So the decoys are actually still from my old thing. So I think that may be my issue. So what I'm going to do is I'm going to regenerate the decoys. And yes, we have something in the project to, so it says, yeah, we're going to get rid of the existing decoys, and I'm going to regenerate them because that will give me all the same transitions. I actually looked at a data set created by someone where the decoys had been left, and all the decoys had had 10 transitions, and all of the targets had 6 transitions. And so then the model had a great time. He said, wow, these targets are really different from the decoys. But the false discovery rate was huge. So you really do have to make sure that your decoys match your targets well. And in this case, so we're now gonna have a message. If you do the kind of modifications that I did and then just try to regenerate your model, it'll say, you know, your decoys don't really match your targets. So to recover from that, I would have to then re-import again. So let's try that. Hope that we now come out with something that we can get our full model. But yeah, that was a good case of don't just keep going. Oh, well, I guess, you know, like, oh, retention time's disabled. That means there's a problem, and go back and look at what the problem might be. So I'm gonna, while we import again here, I'm gonna go back to questions. Instead of spiking IRT synthetic peptides, can you generate an RT model with the highest scoring peptides in your library? And, you know, that's based, yes, you can, and we do, and that's, yeah, that's more advanced functionality, but Kaipo has been working pretty hard on that in the last several months. Some of that functionality is in this version 20.1, and some of that functionality is in the latest Skyline daily. So yes, we're working more and more on just using endogenous peptides, but, you know, still think there's a value to having injected standards. If I generate my DIA spectral library using data from 2 different fragmentation modes, HCD, CID, will PROSIT know to take into account fragmentation mode in their prediction? I think PROSIT is all HCD. Yeah, and that's pretty much for DIA. That's what's, you know, that's what we're interested in. DIA, we'd never use CID. But I don't, you know, yeah, I don't know all of the settings on PROSIT. Can it actually just also do CID spectra? I don't know the answer to that. So you would have to go look at the PROSIT site. Peak scoring models, what influences when score is disabled? The one time I ran my test samples, it did not have all the scores available. So yeah, we'll look at that. If you go to the advanced, so Skyline has a lot of tutorials, and if you go to the whoops. So if you go to the Skyline website and the tutorials page, there's this advanced peak picking models really describes this in detail. It describes, you know, how to deal with, so here you can see a gray line at the bottom here, that's a disabled score, how to deal with that. It usually means that for some reason Skyline couldn't calculate a score for everything. So Skyline really wants to have a score for everything. So I've seen library.product get disabled when there were things that only had spectra that only had 1 or 2 peaks, so only 1 or 2 transitions were used. So let's close that. So there's a number of reasons things can get disabled. I guess if we are stuck in that situation again, I can go through it. So I'm adding a new, and now you can see retention time difference is enabled. But one thing you can do is you can use these. So if I go here, I can see this feature plots. If it's gray, there will be an unknown column. So it'll show you all the things with unknown scores, and you can click a little button to get the list of things that don't have the score, and then you can analyze why that score didn't show up. You can also just, go up to the Skyline support board and let us help you. So, post something with a screenshot saying, why are these scores disabled? And we're more than happy to jump in and try to figure it out with you. Okay, so now we can retrain the model and see how the model looks with, is library intensity dot product still a good score? Yeah, it is. So it's still the strongest score. Shape is actually now a good score. Yeah, coelution count. It doesn't change the scores all that much other than shape being a nice score. So for some reason, shape ends up being better with the PROSIT spectra. Retention time difference is still pretty good, but we had to widen our window to 20 minutes, whereas if we'd kept the old one, it probably wouldn't have been as good. But so yeah, so you can see that worked fine. And now, we have no experimental data. We just did this all based on PROSIT predictions, and we still are doing quite well. So I think that's where I'm going to leave this. Let's see, we'll leave it at 12:30, maybe a question or two more. So I just answered the score, whether the score is available or not. Actually, I can very quickly go back to this advanced peak picking model tutorial, which has a PDF, and I can show you when something is missing. So this is all described in here. So yeah, when something is missing, the graph will look like this. This is a feature score, and this is the library dot product. So in this tutorial, we'll even show you a place where, hey, that not everything has a library dot product, and then you can see this unknown column, and there's a little binoculars button that allows you to then create a list of all the things, and then you can go and assess why they're problematic and why you're not getting scores for them. And usually it's something you want to fix. So I don't see the link for the tutorial we should do tomorrow. When will that be added to the online doc? Oh yeah, so there's not going to be a pre-tutorial for tomorrow. Tomorrow is going to be highly bleeding edge. I'm still working on exactly the steps and what we're going to go through. We are going to be treating fairly closely. I'm just going to cancel out of here. We're going to be working on using the command line a fair bit. So Skyline has a command line execution mode, and when you really want to do a lot of processing, or yeah, you want to set up things to get a better idea of your data and do some trials, you would want, this is extremely powerful. So this is the documentation for the Skyline command line and all its argument values. The original May Institute is sort of for our programmers and people who have some facility with a little bit of programming. This is just batch file programming, but I'm going to show you how to do to use this command line version of Skyline tomorrow. And so I don't have a fully written tutorial for that. That is definitely something that where we would be working very closely with you at the May Institute. And so, yeah, not everything we presented at the May Institute was written up as nicely as the tutorial that was with today's presentation. So is there a way to use PROSIT if one is behind a firewall blocking the connection? There really isn't. Go someplace, get a laptop and go someplace else. And I mean, you can use PROSIT by generating a library. So, go home, generate the library, and then bring the library back in. So at this point, like, I have a document with a Prozit library. Like, I don't need to be connected to Prozit. But there's no offline way for Skyline to just do this stuff. It would require a GPU. So like, there's some possibility that we could create a library that anybody who had a GPU might be able to use Prozit predictions locally, but at this point, there's no local option. Okay, so that's it. So that's it for questions. We're out of time. We're over schedule. Thanks very much for coming. I will stick around for another hour for anybody who wants to actually do this tutorial, not that one, but if you want to, and you got to do it sometime. And if you were at the May Institute, we would just make you do one of these. You can choose the Q-exactive or the triple TOF. I just did the triple TOF data, so it's going to be the most familiar. And yeah, you just go in here. There's a full written tutorial, lots of steps. Here's the download link to get the data if you don't have the data yet. And I'll be here to answer questions anybody might have. So you can just keep posting the Q&A as you do this tutorial. And it will be, if you were at the May Institute, there would be instructors sitting in the room, and we would ask you, okay, now it's time to start doing the tutorial. So if you're up for that, I highly recommend it, and I'll be here to answer questions. Thanks very much. I will try to put something on, there's a question, anything to prepare for tomorrow's talk? I'll try to put something up tonight. I think for the most part, not. People are gonna have to, it's gonna have to be a live thing, not a lot of prep for tomorrow's talk. So if you haven't already done this tutorial, great prep for the talk is to do this tutorial. All right, somebody says they don't understand the point of Prozit. Well, the point of Prozit is that so yeah, I've just used it to replace a library I already had and prove that it works well. But in the case where you don't have that library, and especially if you have a set of targets, like so at this point, I've done this proteome-wide thing, and I'm telling you that, yeah, just asking Prozit to predict. So, I mean, some people have used this to good effect, but my feeling is that asking Prozit to predict all tryptic peptides in the proteome is not a good way to start your experiment. But if you had a targeted experiment, Skyline certainly supports targeted experiments, and we're very want to continue supporting targeted experiments. You have 8 proteins you want to target and you don't have any library data for them, then you can easily use Skyline to use Prozit to predict everything you need to just go out and measure those things. So for PRM, it's a great
everybody leaves, yeah? I just want to also thank Brendan for the presentation, and we have a lot of thank you notes here and also in other emails. So thank you very much.
Thanks to everybody for coming. And yeah, I guess as long as Mina and Olga will leave the connection open, I will just sit here and answer questions.
It is wide open for you for another hour, no problem at all.
Okay.